year. Jeff completed an undergraduate degree in genome science and technology at the University of British Columbia, where he worked with protein design pipelines and programming languages such as R and Python. He used these competences in industry, where he worked for the company Cyclica, which facilitates drug discovery by computational biophysics and machine learning. And after that, Jeff joined the company Structura, which developed software and algorithms for processing of Cryo-EM data. And Jeff has put that experience to good use in his current position as a PhD student in medical biophysics at the University of Toronto with assistant professor Mohammed Mashab Jafari and professor Mitsuhiko Ikura. Jeff's PhD project involves single particle cryo-EM studies of the IP3 receptors. And if anyone's interested, Jeff organizes a weekly online cryo-EM study group with the aim of learning about the mathematics and theory behind the cryo-EM technology. And his talk today is about how one can see calcium ions in cryo-EM data, which I know is no small challenge. So please go ahead, Jeff, whenever you're ready. Thank you.
Thank you for the generous introduction and for all your work organizing the webinars and getting the great speakers and with the junior board. So I wanted to start off with this slide. This slide was taken just a few blocks away from where I am right now, down the street at the University of Toronto in the McLennan building. This is an electron microscope that was made in 1938, and they made it in 4 months. So they worked around the clock with the machine guys making pole pieces and sensitive components in the day shift, and the physics graduate students working the night shift, sometimes till 4 a.m. And it could reach, when they flick the switch, which is, it's quite impressive with all the high voltage and vacuum kind of technology they had back then, they could reach 140 angstroms, soon improved to 60 angstroms, and it worked without any issues for many years. You can read the whole personal account at that link. So it's been a long time since 1938, and just to say that the intellectual landscape of electron microscopy goes back a long way to Ernst Abbe and his optical work. He worked with Carl Zeiss and Franz Zernicke's work on phase contrast imaging. Ernst Ruska won the 1986 Nobel Prize in Physics for his work in electron optics. And he beat this group of graduate students by a number of years, making the first electron microscope in 1933. So a magnified image is formed by individual electrons interacting with the sample and being detected. It's like a double slit experiment, but there's billions of slits for all the spaces between atoms. And over many years, scientists in public and private settings have improved the resolution bit by bit, involving with improvements in precision machining and fast electronics. In the late 1950s, Richard Feynman called for 100 times improvement in electron microscopes. And we've to date only achieved about a 20 times improvement, but we can distinguish much shorter distances than typical bond lengths of atoms, especially in material science applications where the sample is very robust to electron damage. A transmission electron microscope is like a slide projector. You have the thin specimen that must allow electrons to pass through it. And biological specimens are very fragile and have to be imaged at extremely low dose so that they're not damaged. And that makes the images very noisy. The wavelength of a fast electron is more than 3/4 the speed of light in these high voltage machines. And they interact one by the sample and are detected. And they're going so fast that an image forms in a fraction of a second. And so their wavelength is it's in picometers. So that's not a limit to the resolution. The dominating source of noise in biological cryo-electron microscopy is shot noise. It's also called Poisson noise, because the illumination of each pixel is modeled by a Poisson distribution. that depends on the underlying signal. So for Cryo-EM, if we sat on the same area with the beam on, we'd be sort of over here, but that would damage the fragile molecule. And so we have to be more over here and over here. And also, the beam starts to induce motion and damage during the image acquisition, so the underlying signal itself changes and the image blurs out. There's also an effect called the contrast transfer function that introduces optical distortions. The microscope itself does a physical Fourier transform, and this oscillating ringing pattern can be undone by taking a mathematical transform of the image and dividing that out. So just to have a right mental picture, I like showing this video from Gabriel Lander's thesis defense. So you have to, for single particle, you have a pure, biochemically pure sample. You have a membrane protein, you have it in a detergent or in a nanodisk or something like that. And it might be very homogeneous, or you'll have to resolve later on if there's heterogeneity, and you need very small quantities, just enough to get on one grid. So a whole paper can come out of one good sample, or a structure at least can come out of one good sample. There's more than enough particles in that little drop. And there's a lot of optimization that needs to go from this bulk solution to how it's thinned out. And very little is known about the physical forces at that level. There can be issues with calcium contamination, not just during the prep, but all throughout, touching the paper. And then it's frozen, and at that point, it's sort of the biochemistry is done. It's locked in the ice. And you take images and go from 2D to 3D. So the images are projections. Okay, let's go to the next slide there. So here's some real data. This is, as I mentioned before, the ground truth of the 3D structure is not known. You just have these 2D projections. And they're also very noisy. So this is the sort of underlying quality, typical quality. So how do you go from 2D to 3D? You can start off with a sort of blob, and then more or less of the right 3D size of 3D structure, and then estimate how each single particle aligns to that 3D estimate, and reconstruct a new estimate, and then go back to the single particles, re-estimate the alignments again, update your 3D map until those converges and the alignments don't change. Noise in a 3D reconstruction really depends on the relative intensity. So here's, this is actually I don't know if Omid, you recognize it, this is one of your structures you published, and of ranitidine receptor. And it's just thresholded at different intensities, and you can start to see some kind of solvent noise or some noise appear here until you have a cube of noise. So it really depends on the relative intensity. Resolution, since there's no ground truth, resolution, which is quite hot topic in the field, and there's lots of spirited voices. An approach for putting a number to the resolution is to split up the data into two groups, independently analyze it, and then compare them to each other. And you'll have characteristic, this is correlation, and this is the level of detail. And here you can see this would be up here at very low resolution, going to higher resolution, and so on. And eventually the two maps would be very different from each other, and it would drop off. And then you just report the number that it drops some threshold, or give the whole curve. We know a lot about the physics of the microscope and detectors in detail, and so we can simulate what those images look like. So this is the sort of math that's involved in coming up with simulations of also the noise, the shot noise, and the issues with the blurring out of individual electron detection events, because the electron, once it goes in its right pixel spot, it moves around and deposits energy and blurs out. The direct electron detectors, they capture these sparse frames, hundreds or thousands of them, and by summing them up, you start to see your object, but there's the movement in between those sparse frames, and that can blur the underlying signal of just one projection. And then people commonly assume Gaussian additive noise. So the problem is sort of to get at this model, unobserved model, is formulated as an ill-post inverse problem with this imaging operator representing what's happening in the microscope, including the contrast transfer function, the electron optics. The noise, sort of all bundled together, shot noise, electronic noise, is modeled as additive Gaussian noise, which can get quite far. And the actual experimental data doesn't even look like this. There's even more noise, but I just wanted to have you see that image. So, okay, calcium. If there was no noise, could we see calcium in a pixelated and blurry Cryo-EM image? Yes. The pixels are small enough. There's enough signal spread out from calcium ion that's big enough. Yes, you can. But there is noise. So if there was even more blurring, yeah, the blurring would come from beam-induced motion. Can we see calcium ion in a noisy Cryo-EM image? No, I don't think so. Not really. Here you have some noise in the background. You can't see it because it's so small. And this is with calcium. This is a small calmodulin molecule with 4 calcium ions. This is without calcium, and this is the difference. So just the signal just from one atom is kind of starts to be swamped out by the background. But we're really not over here. We're not even over here. We're more over here. And so the signal from calcium ions are completely swamped out. But at the same time, yes, we can resolve calcium binding sites in high resolution Cryo-EM structures. And I guess to almost finish, I wanted to kind of set up a bit of discussion and questions that I'm interested in. So how do you assess that? How really do you assess that this calcium is there? These are some examples from, this one is a heteromeric amino acid transporter, and this is bestrophin 2-calcium activated chloride channel, and they're solved to quite high resolution for the the type of samples they are, and you can see intensity around where they modeled in the calcium ion. However, for sure, there's published structures with calcium in the published atomic model that are not always unambiguously supported by the density map. Sometimes they're, I don't know why they put them in there because I really don't see them. If you want to go into specific examples and take a close look, I'm happy to kind of have that discussion offline, get your opinion, especially if you're a structural biologist. But to assess, you can look at the local resolution, which gives that Fourier shell correlation in a small box, maybe 10 angstroms cubed, around where the calcium ion is. But that's in the kind of whole environment. There's also a confidence map that compares a blob of calcium ion to the background solvent noise, and it gives a green light to regions of the map that pass a stringent threshold. There's also Fourier shell correlations, where you don't compare 2 half maps, but you have a map from a simulated map from a model to your experimental map, and that will depend on evaluating this around the calcium ion depends on our ability to simulate the map of a charged divalent cation like calcium. And to my knowledge and my understanding, simulated maps really don't take into account theory for deep enough to look at calcium ion coordination. Although you can look at the surrounding geometry of side chains and infer that indirectly. So there's a lot of room for improvement for simulating calcium in Cryo-EM. In the code that's in the data processing software that everyone's using, this is an example of how the intensity around a carbon atom that goes to, let's say, relative value of 2.5 and spreads over a few angstroms, it's much more for calcium, up to 10, and it's spreading more. And that's important when you're looking, for instance, here's a calmodulin with some bound calcium. And when you simulate maps, you need to use different values for bound calcium for their intensity, otherwise you really won't see them. And here, this is sort of just using the sort of same background as everything else, and it's not there, and this is with that kind of proper intensity modeled in. And I wanted to show one more thing, which is, let's see here. So the thresholding of the value can tell you a lot. So here we see this is where calcium is, and if only half the particles have calcium, so there's sort of 50% occupancy, you can still see calcium in that area, but the threshold is much less. So metrics that take into account the intensity are very important for calcium because it really stands out above the protein background. And that's it. Thank you very much.
Thanks a lot, Jeff. That's a really nice and different, more methodological overview of the Cryo-EM technology, and also a little sneak peek into the difficulty of visualizing calcium ions in these data. So I think because we're not a lot of people, if someone has a question, you can just raise a hand virtually or open your microphone and ask the question. You can direct it to Jeff yourself. I think that would be nice. So I have a question to begin with, Jeff, because you talked about like having a good sample. So what is a good sample, and how long do you think it takes to optimize a sample to be good enough to get high resolution structure and like concentrations? Question from a non-structural biologist.
Yeah, I mean, a good sample for a Cryo-EM project is one that gives answers a biological question. So that might be more than just reaching a high resolution. You might be interested in some dynamical questions. If you want to reach high resolution, you need a self-consistent set of particles. So you want them to be frozen. You might need to gently cross-link. You might need to throw out a lot of your data and find a subpopulation that's very self-consistent. And in terms of sample preparation, you want to have a lot of particles. You want to be able to get a lot of particles on your grid. So if you're having issues concentrating your protein because it's a tricky membrane protein, you can try special on-grid. There's sort of new on-grid purification approaches. You can put your tag on the grid or have something that attracts your protein. You can apply it multiple times if it's robust enough to survive those multiple applications. Really, you throw everything in the kitchen sink at it and go with what's working. The vitrification process itself is quite heterogeneous. It's not always that reproducible. So within areas around the grid, you have to look and get a consensus. Yeah, it can take years.
Yeah, I can imagine. So I see that Omid has a question. Please go ahead and just open your microphone, Omid.
Hi, Jeff. Yeah, thanks. Obviously, as a recent Cryo-EM convert, I love these types of talks. I just had a question regarding the seeing calcium slide. Obviously, you said you can't see them in noisy images, but I just want to make sure I understand it correctly. Those images that you're showing that you can see calcium, these are average particles or just average
No, this
micrographs?
is one simulated. So this would be the equivalent of a very clean 2D class. This
I
is
see,
just a
I see.
simulated projection with super minimal noise. So if this signal is like varying between 0 and 1, the noise is like 0 to 0.1 or something.
I
And
see.
it's
And
just
there's
very
1,000
small
particles, or
fluctuations. But the calcium itself, which is present here in the projection, there and there, and absent there and there, I mean, it's not hard to see it. And then taking the difference of these two projections, it pops out. But it's almost dominated by this very minimal noise, because the signal that one atom contributes is quite small. And when you have a higher floor of noise, it's pretty minimal here. You can't even see it. Here you can just start to see some speckled noise. Here you can see a lot, but you can see it here, can't see the calcium here. It's really washed out.
yeah, it'd be interesting to see if this can be done experimentally, but I assume there's just simply too much noise in those images. But yeah. Do you know of any methods where people have done two different scanning methods? Because for electron microscopes, you can actually use a different detector to measure some of the maybe inelastic events, and then
Yeah, that would be
contribute that
interesting.
to identifying calcium sites.
Yeah, yeah, you can. Yeah, I'd be interested in people that are kind of trying to go down that road. If you try to leverage the fact that calcium is quite different, right? Its atomic number is 20 compared with 6 and 8, and if you have gold clusters, you can definitely see those in EM images. They're strong scatterers, and negative stain imaging relies on the fact that the scattering if it gets close to a heavy atom, it really goes out and it can not get refocused. It gets caught by the aperture and can make a big signal with What about calcium surrogates? Good question. Good question. Yeah, if they were higher, I don't know much about this calcium surrogate. I mean, if they bound in the same area and it was a larger atomic number, that would sound like a good road to go down. You have to probably, it would be wise to look at some of the maybe chemistry literature on how its coordination might change. There's good papers on comparing calcium magnesium and other divalent cation zinc, so they're not all the same.
Cool. Thanks so much, Jeff. So I just have one other question. So how long does it actually take to? Can you mute your microphone? Just while so how long does it actually take to, obtain the data, and then compare it to how long it takes to deconvolute and refine the structure? I guess it all depends on which kind of resolution you are going for. But could you just maybe comment a bit on that?
If you're doing some screening, you'll have your grid, and then you'll go around and look at different areas, and you'll really be interested in the whole quality of the ice. You're not going for high throughput and capturing many images. So you'll spend an afternoon, a day collecting images, and then maybe spend if you have 50 to 100 images and 10,000 particles or 2,000 particles, you can do that, analyze that in a day and make some conclusions to see if you should change some of the conditions or change your prep. When you go for a high throughput data collection session, that's automated, and so you can monitor it, you can walk away, you can you can sit there and analyze it live. So there's software now that you can do live analysis and see what quality of ice is good and change some of the microscope parameters on the session. The sample's there, but how you're imaging it might change. And then when you want to have and publish pipelines, they're done through, they collect, 5,000, 15,000 images that can be multi-day acquisitions, depending on the speed of the detector. And then there's porting in between programs, redoing second try, third try, to find this self-consistent set to reach a high resolution. But sort of once you have a pipeline figured out and you add up all the times, it might not be that long. But there's a lot of searching through for all the kind of good particles in this soup of heterogeneity or damage or not actually a particle. Yeah, you can play with it for days and days and days, weeks. And in the end, figure something out and do that in a few hours.
Right. Thanks so much, Jeff, for a very nice talk. And I think we will go on now to the next speaker. Omid, can you just share your presentation before we go on? Great.
Can everyone see my screen? Sorry.
Yeah, it works.
Okay,
it works
excellent.
perfectly, Omid. You can just now it doesn't not anymore.
Yeah. Sorry, because I'm sharing my entire screen. That's why.
Yeah.
Sorry, I have to mute myself. Otherwise, you're gonna hear yourself speak. That's why I'm muted.
Okay. Well, that's no problem.
Okay.
I'll try to ignore it.
Okay.
Go ahead. But thanks so much. Thanks for considering it. Okay, so let's go on. So the second speaker today is Omid Haji-Ghassemi, who I had the pleasure of meeting first time at the Gordon Research Conference on excitation contraction coupling in Italy in 2019. Omid grew up in Norway, but he moved to Canada after finishing high school to pursue career goals, and he completed an undergraduate degree in microbiology from at the University of Victoria. And Omid stayed at the University of Victoria to do a PhD in biochemistry with Professor Stephen Evans, where he focused on structural characterization of antibodies in various contexts. And after completing his PhD, Omid continued to work with Stephen for a year before continuing his postdoctoral training at the Department of Biochemistry and Molecular Biology at the University of British Columbia with Professor Philip van Petegem. And during his postdoc, Omid has focused on gaining insights and mechanistic insights into myopathies from a structural perspective, with a focus on ion channels and in particular the ryanodine receptor. And today he will talk about some of his more recent work on determining the structural basis for insecticide modulation of the ryanodine receptor and the inconvenient development of insect resistance. So please go ahead, Omid, when you're ready.
Thank you, Malena. Really appreciate it. Thank you all for coming. This is actually really exciting for me. This is my first talk at the European Calcium Society. In fact, first meeting I've attended. So I'm really excited to be here. And with that, obviously, the ryanodine receptor needs no introduction, but I'll give a very quick overview and then go on to talk about why they're such good targets for insecticides and how we have figured out how this particular insecticide that I'm talking about is binding to and regulating the ryanodine receptor. So as you all know, RYRs is the largest known ion channel. It's made up of about 5,000 amino acids. And there's 4 copies for a fully functional channel, so it's a tetramer. There's 3 major isoforms in humans. There's RY1, which is the major skeletal muscle isoform, RY2, which is in cardiac tissues, neurons, among others, and RY3 is more ubiquitously expressed in different tissues. And so these giant ion channels are, of course, involved in calcium entry from ER or SR stores into the cytosol, and they affect many different physiological functions, from memory formation to muscle contraction. And they're regulated by hundreds of different molecules, small molecules, ions, proteins, and so there's a whole host of different interactions that I think they're very exciting to characterize. And so I've been focusing really a lot of my postdoctoral research on characterization of different proteins and small molecules that regulate this ion channel. And of course, today I'll talk about the clonantraniliprole insecticide. But first, I just want to give a very quick overview of the structural makeup of the iranodine receptor. And this is, of course, work by many different groups, Ning Yang, Ruslan, the Columbia group, and Ranzalk here. And so I'm going to show a dimer. So this is first the transmembrane region made up of 6 different helices, although we believe there's 8. Made up also by something called a voltage like domain, which is it's not really a voltage sensing domain, but it's a pseudo voltage sensing domain. The scaffolding region, which is involved in really this bending of the scaffold beautifully, is coupled to the core of the channel. And then there's a whole bunch of ancillary subunits or globular domains, which are also involved in regulation of the channel, either through protein-protein interactions or protein-ligand interactions. So the project was actually something that was initiated by a former postdoc that was in the lab in Philip van Petegem lab, Michael Yushi, who is now a professor at Tianjin University. This was a really beautiful collaboration between our labs. So he sent 2 scholars, Rufang Mao as a graduate student and Dan Mao as a postdoc, who came in at the university, and I trained them how to prepare the protein samples and how to do the grid preparation for Cryo-EM. And my role of the project was basically do the Cryo-EM processing and some of the modeling. So why ROIRs for insecticides? Well, of course, as many of you already know, the iranodine receptor gets its name from this compound here, iranodine, which is also traditionally in South America used as a insecticide or preparing, keeping food from being infected by different plants or different, organisms. The diamide class of insecticides, which I'm showing here 2 examples of, they are by far the most ubiquitously used in many different countries. They're actually used on pretty much any green plants, and they protect a whole bunch of different crops. There's $1 billion industry, and so it's still very heavily used today. In fact, it's used so heavily that there are many cases of resistant insects now to these insecticides, and they're thought to bind to in a similar way, although that's yet to be shown. So insect ROIRs, they only have 1 isoform, which is in a way makes it a good target. It also has a very low sequence homology. Now, of course, you might think, well, insects don't have a lot of muscle tissue. What is the role of ROIR in insects? We don't really know for certain at this point, but it could be something to do with feeding and general motility of the insects, but we don't exactly understand how that works. The affinity for this drug, clonantraniliprole, or I should say the EC50, is roughly 25 nanomolar. So it's really quite specific, and it is about 250 folds lower in mammals. So that's partially because of the low sequence homology between mammals and insects. And it is known already before we started this project that this compound is increasing the open probability, which leads to feeding cessation, a lack of mobility, and eventually death of the insects. So the project's goal was to first determine the binding site. And of course, there are different R groups within this compound. So maybe we can optimize the binding so that it's more specific to insects, perhaps even alleviate some of the mutations that are found in nature to get around this resistant mechanisms. And yeah, so that was sort of the major goal of the project. And at the time that we were doing this, there was many resistant mutations located in the transmembrane region. So we already kind of knew that the binding site must be in the transmembrane region. However, there was 2 mutations near the SPRI domains, which are located over here, and that made things a little bit more confusing. So we weren't sure if it's going to be multiple binding sites or just the one. And just to show you a little bit how I've calculated some of the EC50s for mammalian RYR, because we knew it already binds from different papers, we could actually, we had a system here that was actually done in Michael's lab in collaboration with a group in Japan, but they made a expressing cell line, hex cell line, that expressed this calcium sensor here in the ER. And of course, as you release more and more calcium from the ER, the fluorescence, the relative fluorescence decreases, and that's how we obtain the EC50 for this compound. So this was not work done by myself. This is all done in Mike's lab. And we were very quickly able to figure out where the drug binds, at least initially, from a very low resolution map of the rabbit RYO1 in complex with chlorantraniliprole. And this is showing you the map in purple, and in blue is the structure from this PDB, which is an open form of RYO1. And we saw that was a really nice fit. So what was clear that in this particular structure and under these conditions, normally you would expect a closed channel. And this is done by many different groups. However, we saw that almost all the particles were open. So that already gave us hints that, okay, there's something going on here. Something's very drastically different. I managed to narrow down the difference to this blob here, of course, at 4.7. And again, this is overall resolution. So its local resolution is probably slightly better in this region. You can kind of see a little bit of side chains. But what we could see here was this blob, and there's clearly many structural changes. Like these residues are clearly shifted, as well as the S4S5 linker here is also slightly shifted. And this got us very excited. As many of the ion channel aficionados know, the S4S5 linker is directly coupled to the gating of ion channels. Now, of course, this is how voltage sensing occurs. And so we already knew that this must be the mechanism. Not only that, but we could also map out some of the resistant mutations. I'm not showing all of them, but one of the major ones, it's a glycine, which is actually conserved between mammals and insects. And this glycine mutated to a glutamate, which you can clearly tell it's probably going to be occluding the binding site, severely impacts the binding of the drug. Similarly, this cysteine here, which is in mammals is a cysteine, however, in insects it's actually an isoleucine. The isoleucine gives the drug much higher affinity, which again makes sense. The drug is highly hydrophobic. It improves the contacts. However, resistant mutation to methionine, again, making that longer chain, longer side chain, preventing binding to the this particular site, and therefore reducing the affinity for that site. So we were quite excited about this, but I still really wanted to know how the drug is bound, because that blob doesn't allow us to discern the different conformations that is possible. So in order to get a much higher resolution, we added a whole bunch of activating agents. So we added ApoCam, this is CAM 1234, and we added ATP, caffeine, and the idea was lock the channel in an open state so that then we can obtain a much higher resolution rather than having a very flexible molecule, because we know, especially when you add CAM, it stabilizes the structure somewhat. And in doing so, we were able to get a 3.8 angstrom overall structure, which I'm showing here in white. And we will also drastically improve the resolution of the binding site, which I'm going to show in a second here, which is located here right next to the S4S5 linker. So we're very, very happy about this. But yet we still had some trouble fitting, although now we were able to orient the drug, we knew which way it was oriented. We still had some trouble figuring out the exact location. You can see here the electron potential map for the bromine is not very good, for example. So we needed more trickery, and we did some local refinements and masking, and so computations I won't cover here today, but basically from using those methods, we were able to further improve the resolution. So I hope the video stopped. And from there, we were able to get a much better fit for the chlorantraniliprole compound onto the binding site. The one unfortunate aspect of this binding site is actually there's a unknown helix, which is so far characterized as unknown 7th alpha helix, and it's actually located really close by. So we think that this helix is probably making interactions, but unfortunately we're unable to map it. And that is this helix here I'm talking about. So again, showing the local resolution now of the binding site. You can clearly see this arginine very clearly making a cation pi interaction with this phenyl group here. And the local resolution is roughly 3.2 angstroms, which I think it's nicely correlates to the map. So, okay, this is great, but what about the mechanism? I haven't talked too much about that. So if you look at the closed structure, so if you take a closed structure shown in green here, this is from a previous RY1 rabbit structure in a closed state. And if you dock onto the insecticide in this location, you see that there will be a steric clash, particularly with that bromine group. And so therefore the S4, S5 linker has to be in an open state for the drug to bind. It cannot bind in this way. And so we believe that that's a mechanism of how it stabilizes the open state of the ion channel. And I can show that again through a morph going from the closed state to open state, where you can see this this S4, S5 linker being sort of pushed sterically by the positioning of the drug. And this is, of course, a very familiar mechanism. I'm sure many of you are well aware of this. As I alluded to before, voltage gating works much in the same way, the sliding of the S4-5 linker. But also there's a compound such as this guy here. I don't know if you guys know what this is. It's the active ingredient in chili peppers, it's caspation, which of course binds to the TRPV1 channels, and it activates the channel in much the same way by allosterically altering the positioning of the S4-5 linker. So just to conclude, the drug cholangioline binds to the pseudo voltage sensing domain. We can map the resistant mutations that either sterically block or weaken interaction. I didn't show examples of that, but we have done experiments and did the same fluorescence readings with mutated channels to show that they actually do weaken interactions with cholangioline prol. And the drug cannot bind into the closed state of the channels. The drug traps RYR in an open state, hindering the S4-S5 linker to slide back and closing the channel. But more interestingly, this is something that we're looking forward in the future is, is this site a druggable site generally? Can we use this site to perhaps look at, other channel openers or that we consider for loss of function mutations as potential therapies? Obviously, you have to be careful here as a lot of loss of function mutations actually because of downregulation of the channel and not necessarily due to mutations. So it'll be a very specific application, but nevertheless, it's something to think about. And can we also design a molecule that could do the opposite, bind to the same site, but actually stabilize the closed state? And I think that would be also very interesting therapeutically. So with that, I want to thank everyone in my lab, of course, Philip here in the center, and I'm eternally grateful for all the different funding agencies that have funded me over the years, either directly or indirectly, in Canada and in the United States. So thank you all for listening, and happy to take any questions. Actually, Danma, this is her second visit, so she's in this slide as well. Yeah, so I'll be happy to take any questions.
Thank you very much, Omid, for the nice overview of your hard work. I was thinking, I do see there's a question from Antonio, but just before, I have a question for you. What was the rationale? Can you maybe comment on that, on solving the structure for the RYY1 isoform and not any of the others? Can you talk a bit about the sequence similarity between the insect channel and the mammalian channels, and why did you choose RYY1?
Yeah, no, that's an excellent question. In fact, the insect RYRs, they have higher sequence homology. I think it's 47% to RYR2, and lower to RYR1, slightly lower, not much, but so the reason we simply picked RYR1 is because it's more stable, a lot easier to work with than RYR2. RYR2, purifying it from source, this is all purified from native tissue. It's tricky to express. It's also very tricky to work with compared to RYR1. So it's just purely a matter of ease. Mike has actually been working really, really hard to try to express the insect RYR. I think he's made some progress, but at the time it looked like it wasn't going to be happening, and we expected competition from two different groups, so we wanted to move forward.
Yeah, that makes sense. And can you maybe also comment on that the local area around the binding site between the different isoforms? Do you expect that this would be transferable to RYY2, for instance, just by looking at structures, alignments?
Yeah, most of the residues, in fact, if I remember correctly, I may be misremembering now, but I think almost all the residues contacting the drug is conserved between RY1 and RY2. So we would expect a very similar binding mode. However, I should stress again, there is that unknown helix that it's unassigned. So if there are differences there, then there will be subtle differences between RY1 and RY2.
Thanks.
But I don't think I think for a follow-up paper, Mike is doing the same test with RY2 as he did with RY1 with the fluorescence reading using the ER calcium sensor.
Right, that'd be brilliant. So we have a question from Antonio Villalobos. He asked, could the chlorantraniliprole pro affect mammalian RYY function? Has there been any toxic effects detected, for instance, on heart functionality?
Yeah, no, this is a really good question. By the way, I applaud you for trying to pronounce the drug's name.
Thanks.
So yeah, so it actually I got sidetracked one day, and I was just trying to pull up some papers and reading, and I found there was a gentleman in I think India who wanted to basically commit suicide by drinking this compound. So this was a bottle that's fairly pure of chlorantraniliprole, and he drank it. And amazingly enough, he survived. Actually, I shouldn't even say he. I don't know if it's he or she. Anyway, regardless, this individual managed to survive, and my bet is the reason being is that you need so much of it to be toxic is because a lot of it gets soaked up in the fatty tissues and adipose tissues. So its actual effective concentration in the blood is not very high, especially if you drink it. I mean, if you inject it, that's probably a very different story. But one of his symptoms was I think he had a very severe heart problem. It was described in the paper. Again, I have to look up the details, but I was just surprised given how toxic you would think this drug would be. Opening up all your skeletal muscle and cardiac tissue RYRs. That cannot be good. But yeah, he managed to survive. He had a heart attack and had some complications long term with his heart. But the problem is when the drug gets soaked up in your fatty tissues, they also get released slowly over time. So you all have for a very long time this compound in your system. But yeah, it's a very good question.
Thanks, Omid. And Jeff has a raised hand. Please go ahead, Jeff.
Thanks for your presentation. I really enjoyed the part where you were showing why you think the resistance mutations are working the way they are, giving resistance. So I wonder if, so a resistant mutation still has to be compatible with a functional channel. So it can't be something that gets the drug out of there but destabilizes the protein or something. So should we be developing tight binders that are specific for just one sequence that's there? Or should we be developing a drug that is more robust for typical mutations that might arise and confer resistance? Or should we have an array of tight binders to anticipated mutations before they happen? Or what are some? I mean, you can learn a lot from the structure, right? So what are some smart ways to really develop good, robust insecticides?
Yeah, I would. I mean, I'm no expert in pharmacology, but my guess would be try to make a drug that is as specific as possible to the various pests that you're trying to target. You also want to avoid insects such as butterflies and obviously beneficial insects. So luckily there are some differences, but unfortunately honeybees, I believe, do get affected by this drug. if I'm not mistaken. They have a very similar sequence, so it's tough to distinguish between the two. So I think ultimately it'd be very difficult to develop a perfectly specific drug that can target just the pests and not some of the beneficial insects. But of course, when you spray them locally, you hope that other insects don't interfere or are not in that local area. But yeah.
Thank you.
Martin has a raised hand. Go ahead.
Hi, yeah, thanks. Apologies if this covers some ground that you talked about. I'm just interested in, so it's similar, so the compound is similar to ryanodine in that it's use dependent, if I've got it right. It binds to an open channel. Does it, but it has a much lower affinity according to your EC50 measurements in intact cells. But the intact cell measurements would have calcium-induced calcium release as a feature of promoting calcium release once you reach threshold concentration. So I wondered if you could comment, because ryanodine has very high affinities, but some in the nanomolar range to lock into a subconductant state that's almost irreversible, it seems. So in this use-dependent manner. So I wondered if the EC50 really did correlate with what you think is the true affinity, and whether there's any evidence for subconductant states that match what ryanodine does to the ryanodine receptor.
Yeah, no, it's an excellent point. And you're absolutely right. That EC50, I would take that with a grain of salt. I don't think that's necessarily an accurate measure of the true EC50. We also don't know if this drug binds to the IP3 receptor. I mean, maybe it's possible. There are some conservation in the transmembrane region, so it's perfectly possible that that's the case. We don't think so. There's no evidence to suggest that it does. But certainly with calcium-induced calcium release, and yeah, we're not controlling it. Because the affinity is not as high as ryanodine that's been traditionally used, then certainly that is a problem. So
there are some curious effects with ryanodine, of course, in that we know that it has kind of a nanomolar affinity, and it's quite hard to wash off. I mean, it's hard to get into the ryan receptor, but you can activate ryan receptors with caffeine, and then it jumps in really quickly because it's got this use-dependent function. But sometimes people have seen effects of ryanodine in the high micromolar range as well. So, and I've never really quite known what to make of that because it's so way beyond what its measured affinity is when you really look at the molecules involved.
Yeah. The thing that's a bit bizarre is a lot of these compounds, I find it strange that they almost a lot of them tend to have biphasic relationships with ryanodine receptor. So that's something that's kind of perplexed us. But we're following this up with plain and lipid bilayer experiments. Obviously, this would be a different system, so that'll be at least more isolated and more controlled system. So
yeah, that'll be really insightful. Great, thanks a lot.
No problem.
Okay, thank you so much to both you, Jeff, and you, Omid, for agreeing to share your work here at the ECS webinars. I will clap for all of us. Applaud. Thanks very much. And I think with that, we will end the formal part of the webinar here. And both Jeff and Omid has agreed to stay for a bit if any of you guys have